Naked mole rat. Underneath the dry soil of East Africa, something that looks like a wrinkled thumb with teeth is living in complete darkness. It breathes recycled air. It shares a tunnel with hundreds of its kind. It will never see daylight. And yet this animal almost never gets cancer. Here is how cancer works. Think of the inside of any animal's body as a busy track, packed with runners moving in every direction. Those runners are cells. Healthy cells follow one rule. When they bump into each other, they stop. Cancer is what happens when one cell ignores that rule. It shoves past everyone and keeps copying itself until the whole system breaks down. The naked mole rat makes that impossible. Every tissue in its body is soaked in a thick, sticky gel, a molecule called high molecular weight hyaluronan that works like foam poured across the track. The moment a cell tries to push past its neighbors, the gel blocks it and forces it to shut down before it can make a single copy of itself. The mole rat's version is 5 times larger and 5 times more common than the version found in mice or humans. The rebellion is stopped at the first step. For every animal ever studied, the older it gets, the more likely it is to die on any given day. In the naked mole rat, that pattern does not exist. A 30-year-old individual has the same chance of dying tomorrow as a 3-year-old. Scientists have already moved the gene that produces this gel into mice. Those mice lived longer and got cancer less often. A problem that consumed decades of medical research was already solved. Underground, in the dark. Greenland shark. 300 meters below the surface of the North Atlantic, no light reaches. The water sits just above freezing. Somewhere in that darkness, a shadow the length of a pickup truck is drifting at slightly less than walking pace. It has been down here since before anyone now alive was born. Before their grandparents, before their great-great-great-grandparents. The shark moving through that water right now was already old when the last great sailing ships were being built. Aging is a fire. Everything alive burns fuel to stay alive, and burning always produces damage. Broken proteins, fraying strands of DNA that get harder to fix the more they pile up. The Greenland shark turned the flame down so low it barely registers. It swims at under 3 kilometers per hour. Every chemical reaction in its body runs in the slowest possible version of itself. Its heart beats at the minimum rate needed to stay alive. Because it burns almost nothing each day, damage builds so slowly that decades pass before it reaches levels that would cause problems in any other animal. Its genome also carries extra copies of the genes responsible for fixing DNA, more repair crews than any other vertebrate, waiting on the small amount of damage that eventually shows up. Scientists worked out the age of the oldest specimen by dating proteins in its eye lens, the only tissue never replaced from the day it is born. Those proteins held a chemical signature from nuclear weapons testing in the 1950s, which let researchers calculate when the shark first came into the world. The oldest one they found was somewhere between 272 and 512 years old. It was alive before most nations on Earth existed. Its eyes contain a chemical record of a world that is centuries gone. Planarian Flatworm. In a freshwater tank at the University of Nottingham, a creature smaller than a staple has been cut in half, and both halves are growing back into complete animals. Each time a Planarian flatworm is split, lengthways, crossways, into fragments. Each piece rebuilds everything it is missing. New brain, new gut, new eyes, new muscles. Researchers who started with a single individual and kept cutting have produced colonies of over 20,000. Here is why cells age in almost every living thing on Earth. Every time a cell divides, the protective tip at the end of its DNA strand gets slightly shorter, like the plastic cap at the end of a shoelace wearing down. That tip is called a telomere. When it gets too short, the cell loses the ability to divide and eventually stops working. Planarian flatworms carry an enzyme called telomerase that rebuilds the tip every single time a cell divides. In nearly every other animal, this enzyme shuts off around birth and never comes back on. In flatworms, it runs without stopping. The flatworm is not resistant to damage. It is resistant to aging because the repair machine never runs out of material. The enzyme that does this exists in human cells too, briefly in early development. And then the switch turns off. Finding that switch is the project. Hydra. In a glass dish in a laboratory, something barely visible without a magnifying lens is extending 8 tentacles into the water and waiting. It has been in this dish, or dishes just like it, for over 40 years. Not a copy, not a descendant. The same individual collected from a pond in the early 1980s. Every cell in its body replaced thousands of times over. Think of a river. The water is always different, New water arriving, old water leaving, but the river stays the same shape. A hydra is the river. Its body runs on 3 continuous streams of stem cells, always dividing, always pushing fresh cells outward, always shedding old ones off through the tips of its tentacles. Every muscle cell, nerve cell, and gut cell cycles through on a schedule of roughly 20 days. There are no old cells inside a hydra, ever. Researchers tracked groups of hydra over 4 years. The yearly death rate stayed below 1.5% regardless of age, a number so flat it looked like a measurement error until it kept showing up again and again. The animal runs the same risk of dying at year 40 as at year 1 because year 40 is, in terms of cell age, identical to year 1. Human stem cells lose efficiency with age. That is part of why healing slows, why injuries take longer to close, why systems fail gradually across decades. What Hydra maintains without end is a stem cell population that never runs down. Turritopsis dohrnii. Something the size of a pinky nail is drifting near the surface of the Mediterranean, transparent, shaped like a tiny bell with trailing threads below it. It is a jellyfish. It looks like nothing special, but this animal is starving, and it has already decided what it is going to do next. Over the next 24 hours, it is going to dissolve its own adult body and grow up again from scratch. Nothing about that is a figure of speech. In every animal on Earth, a cell's identity is a one-way door. A muscle cell commits to being muscle and stays muscle. A nerve cell makes the same commitment and holds it. What Turritopsis dohrnii does when it is starving or injured is reverse that in every cell at once. Muscle cells forget being muscle. Nerve cells forget being nerve. The entire adult body collapses into a blank mass and rebuilds into a young body. Same genetic information, none of the built-up damage. The trigger for this process overlaps with the same genes that scientists spent decades trying to switch on artificially, called the Yamanaka factors, whose discovery won the Nobel Prize in Medicine. This animal uses them in the open ocean, on its own, whenever things get hard enough. Switching a fully committed adult cell into something different is one of the main targets in repair medicine, because it could let doctors replace damaged tissue without surgery. This animal does it completely and reversibly, every time conditions get difficult enough. Researchers are not working from a theory. They are studying something that already does it. Tardigrade. On a microscope slide, something is sitting that looks like a tiny sealed barrel. 8 stubby legs folded inward, body contracted, surface smooth. No heartbeat, no movement, no body activity of any measurable kind. By the standard definitions used in biology, it is dead. It has been this way for decades. The tardigrade is 0.5 millimeter long. It lives in the thin film of water-coating moss on damp rocks on every continent, including Antarctica, including the rooftops of most cities. When it runs out of water, or hits radiation that would destroy any other cell, it forces nearly all the water out of its body and seals into a compressed shape called a ton. In place of the water, it produces a protein that behaves like biological glass, setting hard around every molecule inside every cell, holding each one suspended mid-process. Nothing is happening. Nothing is aging. When water returns and the glass dissolves, the animal releases DNA repair proteins in such numbers they rank among the most common molecules in its body. A second protein called DZUP wraps physically around the DNA strands before damage can reach them, the way padding wraps a fragile object before shipping. In 2007, tardigrades were sent into open space, exposed to full vacuum, extreme temperatures, and direct solar radiation 1,000 times the level that kills a human, for 10 days with no protection. A meaningful number survived. When rehydrated, they reproduced. Scientists have already placed the DSOP gene into human cell cultures, and those cells became measurably more resistant to X-ray damage. The research is still early. Axolotl. In a shallow lake on the outskirts of Mexico City, something is regrowing its leg, not healing it, not sealing over the wound with scar tissue, regrowing it bone, muscle, nerve, skin, and joint in the exact configuration it had before. The process takes a few weeks. When finished, the limb works perfectly. The axolotl does this with legs, sections of its heart, parts of its spinal cord, portions of its brain. The same individual has had the same limb removed and regrown more than 100 times in laboratory settings, and the quality of the replacement does not degrade. Here is why regeneration fails in most animals. When tissue is injured, the wound seals immediately with scar tissue. Simple, structurally flat material that patches the gap, but cannot replicate the layered architecture of what was lost. Once that seal forms, the original blueprint is locked out. The axolotl does not seal. Cells at the wound edge abandon their specialized identities. Muscle cells forget being muscle, bone cells forget being bone, and revert to an uncommitted state called a blastema. This mass reads positional signals from the surrounding tissue and rebuilds exactly what was there, layer by layer in the correct order with working blood supply and intact nerve connections. The axolotl also never fully matures. It retains juvenile features throughout its life: external gills, a permanently aquatic body, keeping its tissues in a more flexible, regeneration-ready condition than any other adult vertebrate. Human cells carry some of the same molecular machinery. The difference is that in humans, it is suppressed almost immediately after early development. The switches exist. They simply stay off. Bowhead whale. Somewhere in the Arctic Ocean, something the size of a school bus is moving slowly through the dark. The harpoon lodged in its blubber, a style of weapon not manufactured since the 19th century, has been there for most of that time. Bowhead whales routinely live past 200 years. Some individuals alive today were born before the American Civil War. The central problem is a mathematical one. Every cell division carries a small chance of producing an error. The more cells an animal has and the more times they divide over a lifetime, the more chances accumulate for something to go wrong. A whale has 1,000 times more cells than a human and lives roughly 3 times as long. By raw probability, every bowhead should develop cancer almost immediately. None of them do. Its genome carries duplicated copies of the genes responsible for detecting and correcting errors in DNA. More backup systems than any other mammal sequenced so far. Its tumor-suppressing genes run at higher activity levels than in shorter-lived animals, constantly scanning for cells starting to misbehave. Bowheads also show almost no sign of the chronic low-grade inflammation that builds up in most aging mammals and acts as a slow background driver of cancer and cellular breakdown. Scientists sequencing the bowhead genome found mutations in genes associated with aging and cancer that appear nowhere in shorter-lived relatives. Those mutations are not damage. They are the result of 200 years of evolutionary pressure selecting, very specifically, for animals that do not break down. The bowhead's genome is a record of what survival at that timescale actually requires, and medicine is only beginning to read it.